Hello students, welcome to today's biology class. Today we are going to understand one of the most important concepts in molecular biology, that is the central dogma of molecular biology. This concept explains how genetic information flows inside a living cell. Simply remember the one sequence, that is DNA makes RNA and RNA to protein. It means that information stored in the DNA is first copied into RNA and then RNA is used to make proteins. So let's first start with the DNA. Let's begin with DNA. DNA stands for deoxyribonucleic acid. It is the hereditary material of almost all living organisms. DNA is stored as genetic information required for growth, development, reproduction and functioning of the cell. You can simply think of DNA as the instruction manual of the cells. Next comes the replication. The first process shown in this diagram is replication. Replication means DNA makes an exact copy of itself. This process occurs before cell division so that each daughter cell receives the same genetic information. Replication is called a semi-conservative process because every new DNA molecules contain one parental strand or one original strand and another strand is the newly synthesized strand. As we know that DNA is the double stranded. So one is original and another one is newly synthesized. This helps maintain genetic continuity from one generation of cells to the next. Coming to the third step, that is the transcription process. In transcription, a specific segment of DNA is copied into RNA, especially messenger RNA, or we can call it mRNA. During this process, RNA polymerase is the enzyme that reads the DNA template and synthesizes a complementary RNA strand. mRNA carries the genetic message from nucleus to the ribosomes in the cytoplasm. You can remember as DNA writes a message and RNA carries that messages. Next, coming to the RNA. RNA stands for ribonucleic acid. Unlike DNA, it is single stranded and there are three types of RNA. Messenger RNA, we called it mRNA, that carries the genetic information. Transfer RNA, tRNA, that brings amino acids to the ribosomes. Ribosomal RNA, that is rRNA, forms the ribosomes and helps in protein synthesis. In this diagram, you can see the RNA sequence is made from the DNA. Here you can see the process of making of RNA from the DNA is known as transcription, whereas formation of DNA by DNA is known as replication. RNA sequence made from the DNA contains the codon, which are group of 3 nucleotides. Here you can see A, U, adenine, uracil, guanine, uracil, adenine, guanine, cytosine. Each codon specifies one amino acid. It means this 3 codon makes one amino acid. For example, AUG, it is the start codon and codes for the amino acid methionine. Next, coming to the process, that is the final step, is the translation. Translation is the process of converting the nucleotide sequence of mRNA into a protein. This process occurs on the ribosome in the cytoplasm. The ribosome reads one codon at a time. Transfer RNA brings the correct amino acid corresponding to each codon. These amino acids join together through peptide bonds to form polypeptide chain, which folds into a functional protein. Here you can see this is the protein translation. Protein, it makes the codon and it makes the smaller protein. Remember, DNA stores the information, RNA carries the information, and protein perform the functions of the cell because they act as enzyme, hormones, antibody, etc. So this entire process is known as central dogma, in which DNA is transcribed into RNA and RNA is translated into protein. So students, this is the central dogma of molecular biology. This topic is extremely important for NET, CUET, PSC and other competitive exams. So make sure you understand each step clearly rather than memorizing it. Thank you for watching. If you find this explanation helpful, don't forget to like, share and subscribe for more biology lectures. Happy learning. Thank you.